Hi there, I'm David DiValentino, and I'm the AIML Solutions Lead and a data scientist at Privacy Analytics. I'm joined today by my colleague Rachel Lee, who is the NLP lead and a senior machine learning engineer at Privacy Analytics, and we're here to discuss building a scalable, machine learning-driven anonymization pipeline for clinical trial transparency. And while our specific use case is healthcare data, the goal of this talk is really to provide an example of privacy protection in practice, and talk about some of the complexities that we encounter translating guidance and literature around anonymization into a production system. Moreover, we hope that the topics that we cover in this talk will provide some utility when thinking about designing privacy-protecting systems, especially as it relates to unstructured text, which is becoming increasingly relevant to many ML-powered applications. So I'd like to start by giving a broad overview of the contents of the talk. We'll start with the definition of clinical trial transparency, then discuss some of the scalability challenges we encounter when dealing with large volumes of unstructured text. Then we'll discuss how one can use AIML to detect personal information in unstructured text, and bring things full circle by talking about how you actually evaluate identifiability of the data subjects encountered in this text. So really the first question that we have to ask is, what is clinical trial transparency? Well, clinical trial transparency is a term that covers the sharing and use of anonymized clinical trial data, either through global public registries or sponsor-supported channels or data portals. And the primary goals of clinical trial transparency include building public trust in medical and pharmaceutical research, as well as driving healthcare outcomes, so essentially empowering researchers to reuse healthcare data to cross-check or validate the results of clinical studies, or even improve the efficacy of their own clinical trials. Now, across the globe, there are many practical guides and regulatory guidance documents describing how data can be used to anonymize data to protect privacy. And our document anonymization methodology centers specifically around regulations covering healthcare data privacy, so HIPAA in the United States, EMA Policy 70 in the EU, or guidance documents from Health Canada. And in particular, the last two documents deal specifically with the public release of information for the purposes of clinical trial transparency. And while specific guidance varies between regulators, broadly speaking, we can consider that anonymized data has some fundamental properties. In this case, personal information is captured in the documents and transformed, and transformations are performed in a manner that renders data subjects non-identifiable. And each regulatory document also provides for the use of statistical and scientific principles to quantitatively assess the identifiability of the data. Now, it's worth mentioning here that most frameworks do have provisions for qualitative assessments of risk, but especially in the case of regulators like EMA and Health Canada, there is now a greater emphasis on the quantitative assessment of risk in this case. So then it becomes a question of, what does it actually mean for public documents to be anonymized? Well, fortunately, regulatory guidance in the EU and Canada has given us some concrete metrics we can use to actually anchor our assessment. Specifically, for the public release of clinical documents, we require each data subject to be statistically similar to at least 10 other individuals, and if this condition is met, we would consider the data set to be anonymized, which is equivalent to saying, in this case, a cell size of 11. So now that we have this high-level definition in place, we have to understand what kind of personal information is contained in the clinical documents. And given the nature and general complexity of clinical data, this creates quite a distinct problem space in terms of the automation and transformation of data. So the personal information that we capture runs the gamut of data types and can be both demographic and longitudinal in nature. So we have, of course, directly identifying information such as subject ID numbers and names and site addresses of study investigators, but we also have to account for the fact that someone's personal information can change over time. So we have indirect identifiers such as age, vital signs data like height and weight, and we also have adverse event information related to the study, which constitutes descriptions of serious and non-serious medical conditions that develop over the course of treatment for a given data subject. So then, how would these quantities look when embedded in a clinical document? So the following text is essentially a marked up example of the type and formatting of clinical information that we'd see in a document. And you can see some common features kind of emerging in this example. Namely, we have multiple data formats, so text can be found in paragraphs or even table formatting. We have several distinct types of personal information present, and they can also be represented differently in the text, so you can have, for example, multiple date formats. We also have fairly domain-specific language relevant to clinical trials. So if one were to actually try to detect the attributes that we'd see and consider in evaluating identifiability, you might see something like the following. Now, in this text alone, we have representations for several different types of personal information. Moreover, it's scattered all around the page with no obvious indicators of the type of a given attribute aside from textual labels and context clues, essentially. So this is really the heart of the problem when dealing with anonymizing clinical text, or really any unstructured text for that matter, because we have information on thousands of individuals, which is very domain specific and heterogeneous, and it can also be fairly ambiguous in nature. In many cases, it's also long-tailed, so we may see only a few examples of a given attribute across 100,000 pages. So given that we have these scale and complexity challenges, we have to ask, can machine learning serve as a possible backbone of this solution? And the short answer is yes. And it's at this point I'd like to throw it over to my colleague Rachel to discuss how we can actually apply AI/ML to detect personal information in unstructured text.
Now, let's take a closer look at how our team has been building AI/ML tools to deal with the practical challenges that we face in anonymizing the clinical documents. So we believe that some of the challenges, some of the lessons we've learned dealing with medical data could also be generalized to other problems from other areas and other domains. So before diving into the technical details, I'd like to touch a bit on some of the major considerations that we had to keep in mind before building the tools, starting with efficiency. One of the lessons we've learned early on is that if our workflow becomes overly complicated, it could lead to dirty data. It could mean that some of the identifiers could be missed, and eventually it could even become an impediment for privacy protection downstream. Another important aspect is to understand our end users. Since our users are not quite equipped with the knowledge of machine learning or data science, we have to build a tool that is user-friendly, even and especially for non-experts like them. So from the perspective of natural language processing, the problem we're trying to solve here is essentially a named entity recognition problem. And traditionally, people have been building systems that are based on handcrafted rules, based on guessed tiers, and pattern matching for this type of tasks. So these systems, for instance, in this case, someone could have explicitly written out these regular expressions to find specific things for certain formats, like these dates or medical histories. So these type of systems are easy to implement. They are also very straightforward to understand. But the problem is with the scale of the problem that we're facing, with the thousands or tens of thousands of pages and numerous layout and format variations, this approach is very difficult to be efficient. This also sets a relatively high bar for users because you have to first possess the knowledge of creating these types of rules. Now, that's why we turn to a machine learning-based approach, hoping to create a more efficient system and also lower the barrier for the users. What machine learning does and is good at is to automatically extract patterns from the training data, labeled, annotated by the humans. Now, the benefit of machine learning over this rule-based system is evident. It could still scale better, and it requires less knowledge beforehand. With that being said, machine learning also has its own limitations. When we try to deploy a machine learning-based system in practice, usually there isn't enough training data to start with, meaning we cannot really train a performant model right off the bat. So with technology like transfer learning, we are actually able to leverage existing knowledge from related tasks or related domains to have a jump start on our own task here. In our case, for example, we could use a technique called fine-tuning to transfer knowledge from pre-trained language models and to apply the knowledge on our own named entity recognition task. Now, through experimenting with different types of pre-trained models, we found that domain-specific language models, in our case, these models were pre-trained with medical data, they could help us achieve better performance comparing to other more generic pre-trained models. So here is what a machine learning-based anonymization workflow would look like. The users would first label a number of documents. With an ML algorithm, we would fine-tune the model and apply it on the rest of the documents that needs to be anonymized. Then, because the model is never going to be perfect, we would still need the end user to eventually examine the results of the model and correct any errors, as the model is never going to catch 100% of things or make no errors. So in a way, if we consider these ML algorithms as the engines, the data really is the fill. Without the data, our system is never going to take off in practice. So an issue when we try to deploy these machine learning based systems in practice, and we keep coming back to, is that we cannot really train our model once and be done with it. So for, let's say for instance, there has been research showing that off the shelf, these NER models, in some cases, they would catch maybe 70% to 80% of the things. While if we retrain the model with the right data, with the right distribution, those models could actually detect over 97% of entities. And in our use case specifically, because any errors that's made by the model is going to be hand corrected, be manually corrected by our end users. So it is critical to make sure that we keep retraining our models when the incoming distribution changes. Then the question becomes, how can we train a model faster? So here's where a technique called active learning comes into play. The core idea of active learning is that if a machine learning system is intelligent enough, it asks the right questions and seeks help from the human whenever needed. At the end of the day, we'll have a system that can learn faster with less training and less cost. So how is it going to change our workflow? Now, our system will start building a model with only a small amount of training data. Iteratively, it chooses the documents that it considers to be the most informative and asks human to correct them. These selected documents are fed back into training to improve the model until the model performance is good enough or it plateaus. When we compared active learning with a traditional learning, or we can call it passive learning system, on the real-world data set, we found that active learning learns in a more stable manner, and it could grow to a certain level of performance faster than passive learning. So if we want to translate that type of findings to savings in training data, we can see that it's possible to save 30% of the training documents if, let's say, 0.9 is our targeted detection rate. We further conducted a user study to compare the active learning system with a legacy rule-based system, and the difference there is even more notable. With the help of active learning, we were able to cut down the total detection time of 100 pages by almost 80%. Now, this concludes my segment on AI ML. We talked about active learning. We also talked about transfer learning. Feel free to leave any questions in the chat. And for now, I'll just hand off to David to talk about identifiability on free text. Thank you.
So at this point, we've come full circle. We've talked about what anonymization means in practice, and we've understood how we can leverage AI/ML to build out a detection pipeline. Now, given that any human or machine-led process will have some finite accuracy, what does that actually mean for identifiability? Well, just to give a brief reminder, for the public release of clinical documents, we typically require each data subject to be statistically similar to at least 10 other individuals, or equivalently a cell size of 11. And so the question is, how can you tell when someone is actually similar to 10 other people? Well, to align with existing regulatory guidance, we first have to define what's known as a reference population, and this quantity is akin to an upper bound on how similar an individual is to people in the broader population, absent any other identifying information. So in the context of CTT, typically we take this quantity to be the number of people that were in similar clinical trials at the time, based on their demography and the condition being treated. So they look like they could have been part of the study, essentially. Now, in defining this quantity, we have to make some informed assumptions about the knowledge of an attacker. For example, would an attacker know precisely the clinical trial that a participant took part in, or would there be a range of possible choices? And these assumptions inform the size of the reference population that we wind up with. So by comparing the information we've detected about an individual against the reference population, we wind up with a numerical value for each data subject. And this represents how identifiable they are. And this quantity is determined relative to the cell size of 11. So in practice, we would express this as a quantity of a disclosure risk measurement by requiring that the risk of re-identification for each data subject must be less than 0.09 or 1 over 11. Now, to the astute viewer saying that these look like confidence intervals, you would be correct, because one thing we haven't touched on yet is how finite detection performance affects identifiability. Now, humans and machine learning algorithms are both fallible. They can miss identifiers or just detect the wrong thing. And so our measure of identifiability has to account for the fact that we may have missed some information. So typically what we do is we ask two data analysts to create a gold standard sample of pages, which are sampled from throughout the document. And based on this manual annotation process, we can derive a confidence interval for recall. And this plugs directly into our risk measurement. So higher values of sample recall translate to a smaller footprint in terms of missed identifiers on disclosure risk. Now, ultimately, in most instances, the data is too identifiable to release publicly, so we have to transform it, either by generalizing certain attributes or suppressing them outright. And you can see an example of this here, in that age has been generalized and replaced with a similar looking value. Some medical histories have been suppressed, some have been retained, and some have been generalized to a more broad term. And we also have an instance of a date here which was transformed to an earlier value based on a date shifting algorithm. So in this regard, we have to choose transformations that are useful for the endpoints of clinical trial transparency, but also sufficiently privacy protective. And so we're balancing a trade-off between data utility and privacy protection while still trying to maintain that baseline level of protection to facilitate the public disclosure of the documents. So just to wrap things up, ultimately we found throughout this talk that document anonymization requires an approach which is tailored to the format, content, and knowledge domain of the unstructured text. And while machine learning and AI can be used to enable scalability in these types of systems, humans in the loop remain a critical element to guide the performance and domain of such systems. And so ultimately, given that any human or ML-led detection system has finite recall, we have to account for this variation when assessing identifiability and ultimately transforming the data for release. And that is all for our talk for today. Thank you very much for your attention.